So I'm going to talk about a few new ideas as to how we can take things forward in Ring 20 research, and why Ring 20 can be an important model for epilepsy in general. And this may be a way, although this is a rare epilepsy, and that's been emphasized multiple times this morning, because of its particular pattern and particular nature, it may actually give clues to epilepsy in general. And epilepsy in general is very common. Epilepsy affects 50 to 60 million people in the world, about 400,000 people in the UK. So although the individual types of epilepsy are not common, as a whole, as a disease, epilepsy is very common. So if we can find any insights from rare conditions into the broader management of epilepsy, I think that's very, very valuable. So epilepsy, learning and behavior are all intimately linked. We often, as doctors, we decide we're going to have a specialty. I'm going to be a pediatric neurologist and an epileptologist. But actually, if you're an epileptologist, you're dealing with the brain. And we talk about all these causes, these epilepsy genes, but in fact, they're not really epilepsy genes. They're brain genes. And Ring Chromosome 20 is not an epilepsy syndrome, really. It's a brain syndrome, because it affects learning, behavior, and all aspects of brain function. So when we talk about learning and behavior with epilepsy, obviously there's a relationship with the underlying cause of the epilepsy. There's a relationship with age of onset, and we see this frequently in all epilepsies. Generally, the earlier onset you have, the more impact it has on learning and behavior, because as you know, any small baby is just learning constantly. And the first few years of life are critical for that learning to occur. So the younger the age of onset, the more likely learning disability and behavioural problems are going to be. We also see a relationship with frequency of seizures and EEG abnormalities, and I'll talk about that. There's a relationship with age and duration of the epilepsy. So if you're 15 or 16, compared to if you're 8 or 9, the impact of learning and behaviour may be different because your epilepsy's gone on for so many more years. There's a relationship with medication, which you all know about. The more medications you get, the more interactions you get. Many individuals with Ring Chromosome 20 are on 2, 3, 4, and I'm sure many places in the world 5, 6 anti-epileptic medications. And the more drugs we give people, the more side effects you're going to get. And the relationship with family issues. So learning for any individual also depends on what their family's like. If you have a family that's really nurturing, pushing, giving you lots of experiences, taking you out, meeting lots of other children, you will learn more. Even if you have a learning disability, you will learn more. So the experience that the family gives the child will influence their learning as well. And with the schooling, how good your school is in many respects. Does your school acknowledge your child's difficulties? Does your school provide you with the one-to-one support that you need? Or maybe not one-to-one, but additional support that you need for your child? Do they understand the way your child learns? And do they give you a program to focus on the way your child learns? So that so many different complicated issues here that relate between epilepsy and learning. And so this is very much a bi-directional relationship. So the epilepsy impacts on the learning, and the underlying cause of both those things impact on learning and epilepsy. So we know that academic achievement in school-aged children with active epilepsy is poorer than in the general population. So we have low achievement in more than 50% of children with epilepsy in schools have lower than expected achievement. And this is all epilepsies. So learning is important in epilepsy. Now, we've heard this term before, epileptic encephalopathy. And I think this is the way in which Ring Chromosome 20 can inform wider epilepsy. So this is the definition of epileptic encephalopathy from a recent paper on classification of epilepsies. So it's where the epileptic activity itself contributes to severe cognitive and behavioural impairments above and beyond what might be expected from the underlying pathology alone. So that means that you've got this burst of abnormal electrical activity going on in your brain. That's going to affect the way you see the world, understand the world, the way you learn. And can that excessive electrical activity then impact on your learning? And we know that that can occur. Also, the key concept here in epileptic encephalopathy is that reducing the epileptic activity may reduce the developmental consequences of the disorder. So, if you can reduce that epileptic activity, can you reduce the learning disability? Now, in many epilepsies, I think we can't do that. Many epilepsies with certain causes, because the underlying cause of the epilepsy, whether that's a brain malformation, the brain was just not formed in the right way in the womb, or the brain has been damaged, that will be the primary thing that influences the learning of that individual. And however much we reduce the epileptic activity, we may not have a big impact on the learning. But in Ring Chromosome 20, we see these children who have normal development often, typically, before the onset of their epilepsy, right through to early primary school years. So they are a group of children and adults who this may actually be much more relevant to than many other people with epilepsy. Because you have a normal child who then develops epilepsy and then gets cognitive and behavioural problems, as we've heard from individuals' experiences this morning. So, I wrote a paper with colleagues in a debate series in one of the big epilepsy journals called Epilepsia, where we argued that epileptic activity really is a surrogate of the underlying etiology, and stopping the activity has a limited impact on developmental outcome. Now, this is one of these medical debate papers where you have to take one extreme point of view. But one of the things I said in that paper is the clearest, in the younger child, is best illustrated in infantile spasms or with hypsarrhythmia, and in the older child or teenager, the clearest example is Ring Chromosome 20. In both of these situations, an individual who may have been previously developmentally normal could develop rapid and significant cognitive and behavioural decline in epileptic seizures. So this is one of the situations where I don't think this is true. So ideally, if we can get the electrical activity and seizures improved, then I think there is a prospect for improving outcome in children and adults with Ring 20. So this is one of my great embarrassments in medicine. And one of my embarrassments is that many years ago I did a lot of work on Ring Chromosome 20 with Arno Biraben, and we collected 50 cases from around the world. And these are the ages of which they present. And this has been presented at big medical meetings, but we've never got round to publishing it completely because there's so much data, and I've never had a research fellow to do that data. Hopefully that will change. And speaking to Professor Canavini, I have some ideas as to how that might change. We can get this all published. So you can see these children present primarily early primary school years. And we know the behavioural phenotype: poor attention, concentration, impulsivity, disinhibition. Some children have obsessive behaviour, some children have aggressive behaviours. And those features were seen in 68% of these individuals. In fact, the majority of French children saw a psychiatrist before they ever saw a neurologist. Different system in the UK. They would usually come to a paediatrician before they saw a psychiatrist, but in France, often seeing psychiatrists first because of the behaviour decline before the epileptic seizures. All school-age children were in normal schooling at presentation, and cognitive decline was associated with epilepsy onset. In our series, the IQ of the individuals didn't correlate that well with age of onset. In the UK cases, and at that time we had 8 UK children, developed an autistic phenotype. So problems with social communication, obsessive behaviours, understanding what other people feel. But overall, if we take the whole group, 34% were not impaired cognitively and described as normal, completely normal. 40% had specific difficulties, and 52% have moderate or global difficulties. So we've still got half of the individuals there who, despite their epilepsy, have normal IQs without even any very significant specific difficulties. So that's important because that might give us clues as to why some individuals do better than other people. And often the answers are in their constitution, their particular makeup, in their genetics, and hopefully we'll hear about the genetics. So, examples of cognitive evaluations within our group. So, you have a full-scale IQ in someone of 86 at 6 years, but it's down to 60 by 30 years. Someone at 93 at age 9, 69 at age 11, and 46 by 12. So this is illustrating cognitive decline. 72, 55, 96, 64, and then someone coming back up again. So it'd be interesting to know why that person came back up again. So if you were seeing this in adults, what would you'd say this is a dementia? We'd use it. We don't use that term much in pediatrics or in children, but effectively that's what's happening. You're losing your skills, you're losing your cognitive abilities, and it can be a progressive process. So this is really important for us to think about how we manage this better. And there's lots of funds and money out there for dementia research, but that typically is focused on the adult population because that's where most dementia obviously occurs. But if we're thinking about funding in the future, then maybe that's a target that family groups and other people, individuals can look at. So we looked at percentage of mosaicism in peripheral blood lymphocytes, and that in this large group varied from 0.5%. So that means 1 cell in 200 had the mosaic ring 20 to 100%. One child was diagnosed post-mortem, and one mother had a bit of ring 20 mosaicism with no symptoms, and that's the percentage mosaicism. And we did a scatter plot of mosaicism and IQ, and the percentage mosaicism correlates negatively with IQ in 28 cases, where a specific IQ value was available. So there is a relationship there. And percentage of mosaicism and age of epilepsy onset, and it correlated negatively with age of onset. So there was a relationship there, although we didn't get an independent one between age of onset and IQ, which is all statistics. And we know that these people with ring chromosome 20 have this non-convulsive status, so with this very abnormal EEG. So if you imagine you're trying to learn anything with this very abnormal EEG, it's very difficult to learn. If you have this abnormal EEG going on when you're sleeping, sleep is the time when your memories get encoded, we think, into your long-term memory. In the daytime, the theories are that we save our memories into our hippocampus, a bit like a USB stick, and then we move it into the hard drive at night time. Okay? And we move it into, and so if you cannot move it into the hard drive at night time, because the EEG is abnormal and it's affecting all this your normal electrical activity, then again, that's going to impact on your learning and behavior. So we have that cohort of so many patients that we looked at so many years ago. Now there are many more patients, and through the ERN that we heard about, and through other groups, and I know that there are other groups. There's a group that a German pediatrician called Gerhard Kluger runs, where he has databases of rare genetic disorders. And there are lots of these databases scattered around the world that could come together, hopefully through the ERN networks. And we might find many, many patients. And through finding many patients and through describing them, a bit like we've shown and other colleagues have shown, then we can get the phenotype and the long-term natural history even better characterized. And I think there is a value in doing that. Obviously, that may not give you a new drug treatment initially, but it might give you clues as to what the important things are to treat in individuals with Ring 20. And just to illustrate what large cohorts can do, so we do testing for another rare disorder that you've heard about called Dravet syndrome, and we were able, because all the patients for the testing for many years were tested in our lab in Scotland, we had 240 patients. We were able to get all the clinical information for, all the detailed EEG data for, genetic characterization, and we could do lots of research on those individuals. So we could look at how their behavioural problems, their autistic features changed over time. We could look at the drugs that worked in those individuals. We could look at the drugs that made things worse. We could look at quality of life in this syndrome called Dravet syndrome. And that work could also be done in Ring Chromosome 20. So I think I'll just move on. So we know that we have focal seizures in Ring 20. We've heard this definition before. So in a focal seizure, you have the onset of the electrical activity, and it stays in one hemisphere of the brain. It might spread to another hemisphere of the brain. But if you imagine this pulsing electrical activity is going on all the time in your brain, it's going to be really difficult for you to learn, pick up things, process things. And we've heard, I think the young guys who are producing this app, so they're all about looking at processing speed, and that's going to be impaired if you've got lots of electrical activity going on in your brain. So can we learn anything from neuro imaging? So Professor Canavini and her group have done some really interesting work looking at brain networks and the contribution of neuroimaging to understanding Ring 20. And she didn't talk about this morning, so if I get anything wrong, she can tell me. So she's looked at the networks involved in the brain in Ring 20. And through very clever work, they can do both EEGs and what we call functional MRI at the same time. So through this, they've shown that it is really the frontal circuits that are involved in the seizure activity. And as you get seizure activity, you can look at oxygen uptake in different parts of the brain, so the function of the brain. And you can look at the networks involved. What they also showed is that a bit of the brain at the base of the brain called the basal ganglia, which has been implicated in Ring 20 and might be important in stopping seizures normally, is also involved in the seizures. So that's producing new insights. Also, there's a network in the brain called the default mode network, which is the resting state of the brain. And the resting state of the brain is abnormal because often when the brain is resting, it's not really resting. It's processing lots of things, it's learning, it's dealing with everything that we've experienced in our life. So this is disrupted by the abnormal EEG activity. So the resting brain is disrupted. So these are important, this work, because then if you then get new treatments and can alter the activity, then you can see whether that functional changes that Professor Canavini and colleagues have shown, whether that improves. So these are developing markers for the actual dysfunction in the brain. This is also showing, I think, some of the basal ganglia involvement at the base of the brain in individuals with Ring Chromosome 20. So this is all really complicated, detailed, functional neuroimaging, which is difficult for a simple neurologist to understand, and we have to work really closely with our colleagues in neuroimaging, in neurophysiology together. So this isn't work that one specialist can do. It has to be done by a team of doctors and statisticians working together. So what are the prospects then for pushing this work forward? So now, as neuroimaging is one of the areas which has expanded massively in medicine. So as well as, so we have MRI scanners. All your children will have had MRI scans, probably on what's called a 1.5 Tesla magnet. 1.5 T is the strength of the magnet. But those magnets are getting stronger and stronger. And what that does, it allows you to look at the brain in even more detail. So now we have 3 Tesla magnets, which are now being used for epilepsy surgery programs around the world. And now we're going on to 7 Tesla magnets, which are primarily being used in research programs. So this gives an even more detailed picture of the brain. So we have, there is a UK 7 Tesla network. So this is a building that's just been built in Glasgow, brand new. That's a photograph I took on a typical cloudy Glasgow day, Thursday evening, before I left the hospital. So no one's actually been into that building yet to have a scan. This is the scanner being lifted into the building. This is what a 7T scanner looks like. But the nice thing about this scanner is here. And this is the adult hospital, and this is the children's hospital. So there are very few of these these 7T scanners on a hospital site. And the reason for putting it on a hospital site was, as well as just using it for fancy neuropsychological research and function neuroimaging, we can put patients into the scanner, people with clinical problems. So we have a very active stroke research unit in Glasgow, very high incidence of stroke in Scotland because of our unhealthy lifestyles. But we have a very a big stroke research team. And one of the thoughts is, perhaps can we get children with ring chromosome 20 imaged in some of these really high field magnets, particularly older children and adults, and see whether there are any very subtle structural abnormalities that we might be able to see. That's a possibility. Or more important even is maybe we can do more functional studies in those children, which we couldn't do on the imaging techniques we've got now. So with some of neuroimaging, you can look at something called connectomics. So you can look at how the brain, parts of the brain are connected together, and you can get these lovely pictures of different pathways in the brain. And we could see, well, are there any subtle disruptions that we see in children with ring chromosome 20? Are there, can we define differences between those, between individuals with ring 20 who've got normal cognition and those that don't? Can we define differences between severity of epilepsy and treatment response? And can we also relate this to genetic characterization and expression of genes? So doing this fancy neuroimaging alongside genetic studies and detailed phenotyping of patients. And that can work sometimes when you have all these things on one site. So when we looked at the group of 50 patients, all patients had a drug-resistant epilepsy. No single medication or combination proved consistently helpful. Vigabatrin was a drug that exacerbated seizures. Several cases that we had surgical evaluations prior to diagnosis, and one patient even had 2 surgical operations before they got a diagnosis, bits of their frontal lobe removed. And that was actually a patient of someone who, and he won't mind me telling you this, who's the godfather of epilepsy genetics over the last 50, Sam Berkovic in Melbourne, one of his patients. And he told me about this later, and he only made the diagnosis after the individual had had 2 operations. So patients had seizure-free or relatively quiescent periods for some years reported, which I wasn't sure, but I wondered whether this was independent of any therapy. So were we actually making any difference with our drugs? And sometimes as doctors we have to be honest and sometimes say, well, the medicines aren't working all the time. The nature of epilepsy is it's like this. And often you start the medicines when the epilepsy is bad. So the epilepsy is always going to get better, usually when you start the medicine. And often parents would say, oh, we always had a honeymoon period. We had like a few months where the epilepsy got better when we started the drug. And so they say that was due to the drug, but actually it might have been that was a time You start the medicine when things are bad, and it's always going to get better. So this is the no consistent response from our groups. Oxcarbazepine response, carbamazepine query response, sodium valproate query response, levetiracetam query response, clobazam query response. VNS response possible in 2 cases, no response in 1 Spanish cases, 2 cases from Belgium, no response, 1 case from the US response, 1 case no response. So no consistency either with VNS at that time. I don't know whether the newer VNS devices may be helpful, and one of the things about VNS is that when people are very reluctant to try medicines because of side effects, it may not have the same types of side effects that medications have as a trial. Oops. So this is what we're all excited about in epilepsy genetics. Precision medicine. And this is when we had an American president that we could all look up to. So the Precision Medicine Initiative in the States, and I looked at this last night, 2 nights ago. Trump had proposed slashing NIH by 1.2 billion, but luckily Congress said no, and they've kept the funding. So that's good. So how can we get precision medicine in the epilepsies? And how can we get it for Ring 20? So we have all these genes, and we've had lists of genes before that we know are there for epilepsy, and you think, well, we've got a gene that causes epilepsy, let's get a precise medicine that targets that gene. And there are lots of potential treatments that can target different types of processes in epilepsy, particularly for the metabolic epilepsies, and for some of the other neuroreceptors there may be specific targets. But in Ring 20 things are more complicated, because we don't have one gene that's wrong. We probably have the ring, and we'll hopefully hear about this in the next talk, maybe affecting the expression of many genes, which may have diverse functions in the brain. So that's more complicated. So for many people, they might think, oh, well, it's too difficult for us to think about. How do we ever think about getting precision medicine? You look at the complicated pathways in the brain, they're so complicated. These are all the metabolic, potential metabolic pathways in epilepsy. What if Ring 20 affects lots of these? How do we get there? This is really challenging. So we've recently seen another ring problem, tale of another ring. So this is a child with a condition called has got a mutation gene called SMC1A, and she presented with a difficult epilepsy, with clusters of seizures. And actually I saw her the first time she attended the EEG department in my hospital. So the technician asked me to come through because Mum and Dad had brought a video to the EEG. So she hadn't been referred to me, but she'd just come for an EEG. But the technician said, Mum and Dad have got a video of the event. Would you like to see it? So I saw the video and said, yeah, she's definitely got epilepsy. And then she was diagnosed ultimately through a UK study called Deciphering Developmental Disorders, which is a really important study where 13,000 individuals have had whole exome sequencing. That's all their coding genes looked at, along with their parents. And we are making diagnoses in about 25 to 30% of people with intellectual disability. And not surprisingly, about a quarter of those people have epilepsy, because these are not just intellectual disability genes, these are brain genes. And epilepsy is a symptom of a brain problem. So anyway, saw this girl with diagnosed with SMC1A related epilepsy. Now what SMC1A does is it helps hold together the chromosomes or parts of the chromosomes during important phases in chromosome division. So it makes part of what's called a cohesin ring. And my colleague sitting to my right will know a lot more about this than I do. But it's a structural maintenance of chromosomes 1A, this gene. So this gene and these cohesin rings have got an important role in chromosome segregation, DNA repair, gene expression, development, and genome integrity. So they do lots of things. So we think if you've got a mutation in this gene, it's affecting probably lots of other genes. So how do we look at that further? You know, because it's a bit like ring 20. So these cohesin rings do all sorts of things. So they're important when cells divide up and produce identical daughter cells, and also important in division in the sex cells as well. So we managed through my research fellow collected lots of cases from all around, primarily from the UK and Europe, and we wrote a paper about the presentation of these children together. And now the next step is seeing, well, actually, you know, can we see what are the consequences of this mutation? So we've got a grant now to look at what's called transcriptomics. So DNA is goes through a process of transcription in terms of producing the protein. And one of the things that's produced is messenger RNA as a product of the DNA and transcription. So if you want to measure actually what genes are being produced or not produced, you can look at the RNA. And there are techniques now to measure that, and we'll hopefully hear about that in the next talk as well. So one of our thoughts in this group, we've got a grant to look at taking skin biopsies, which are just simply 3 millimeter skin biopsies from people with this particular disorder, and we'll plan to look at the expression of their RNA, see which genes are either produced too much or too little, and that might give us clues as to what's happening in the cells. And we've got 15 children from around the world who we can recruit to that study. So we've got a small grant of just £12,000 to do that, and potentially this work, our thoughts could be done in Ring 20, but we might hear that that's already been done. So I think these are the key things that we need to look at. We need to look at detailed clinical characterization with cross-sectional cohort studies of individuals with Ring 20, and we'll get this from the databases that are being developed. We need to use the latest neuroimaging techniques and functional neuroimaging, as Professor Canavini and her group have been doing in Italy, and we need to use the latest technology to define the genetic consequences of ring formation. And those are, I think, the next steps forward. And from those, we may find pathways to hopefully better treatments and better outcomes.